Hello students, how are you? I hope you are fine during these difficult days. So let's start our course. The course is Biotechnology of Microorganisms, and my name is Aleksander Łukanowski, and I provide this course this semester. So let me tell you more about the schedule of the course. The whole course will take 15 teaching hours, and the course is divided into lectures and labs. The lectures concern PCR technique, and the second one will be about real-time PCR technique. After that part will continue with labs. Because of coronavirus situation in Poland, the course has to be provided online. As I know, our university will be closed till 10th of April, and I'm afraid that this break can last longer. And so we have no choice. We have to provide the course online. So let's start. Last week I sent you email with the links to basic information about PCR technique. I hope that everyone read this and successfully passed the short test at the end. Today, when you have basic knowledge about PCR, we can continue with lecture on this technique. So let's start. As you see, the abbreviation PCR. PCR means polymerase chain reaction. PCR is not the new technique. It was invented in 1985. It was 35 years ago. But nowadays we have many modifications based on classical PCR. PCR assay is still used very commonly in the laboratories all over the world. Please remember that PCR is a technique which bases only on DNA, not RNA. When you want to analyze RNA, you have to make special analysis which is called reverse transcription, and this is the preliminary step for PCR reaction. In this reaction, RNA is translated into cDNA by an enzyme which is called reverse transcriptase, and such obtained DNA molecules can be used later in classical PCR as a template. This technique is called RT-PCR, reverse transcription PCR, and is useful for research of plant viruses with genomic material is RNA instead of DNA. So please remember once again, before I start talking about PCR reaction, when you use PCR reaction, you have to take DNA as a template, not RNA. PCR concerns only DNA analysis. And how DNA is constructed? What is the structure of the DNA? DNA is a macromolecule made of single monomers. These monomers are called nucleotides. Two strands of DNA are held together by four types of nucleotides, which consist adenine, cytosine, thymine, and guanine, and they make pairs: adenine with thymine AT and cytosine with G, CG. The strands of DNA molecule are complementary to each other. We have two complementary strands. Sequence of one strand is completely complementary to the other strand of the DNA. You see this on the picture. It means that knowing the sequence of the first strand, for example, the sequence of this strand is ATGCA, we can predict the sequence of the second strand, which will be in this case TACGT. And we can build the new strand of DNA during PCR reaction with the use of special enzyme which is called Taq polymerase. So PCR was invented to amplify in vitro of specific DNA sequences with thermostable enzyme which is called DNA polymerase. And DNA sequence can be selectively amplified in just a few hours, only a few hours. It is a very quick technique and allows us to amplify huge number of the copies of desired fragment of DNA. Polymerase chain reaction PCR was invented in 1985 by Carey Mellis. He was awarded for this with Nobel Prize in Chemistry 8 years ago in 1993. It has become the most widely used nucleic acid amplification technology and standard for amplification processes in diagnosis and research. So here you see Carey Mellis, the inventor of PCR reaction. And please let me tell you more about abbreviation of technique. The name is PCR. R of course means reaction, but what does it mean P and C? P is for polymerase, and why polymerase? It is called polymerase because enzyme used in this reaction is DNA polymerase. The technique was made possible by the discovery of Taq polymerase. This is DNA polymerase that is used by the bacterium Thermus aquaticus that was discovered in hot springs. This bacterium is thermophilic, and the whole enzymatic apparatus of this organism has to be heat resistant. Why is it so important for us? Because the last step of every cycle in the reaction is provided at constant and high temperature, 72 Celsius degrees. Enzyme must remain active at this temperature. That's why only heat-resistant enzyme can be used under such thermal conditions. This is the picture of hot spring in which our bacterium Thermus aquaticus lives, and bacterium looks like this. Is microscopic picture of this bacterium, Thermus aquaticus. And why C? C is for chain. Why chain? Chain because the products of the first cycle become substrates for the next one, and so on and so on and so on. So we can tell that we have some kind of the loop, cycle after cycle. And what is very important about PCR? PCR reaction mimics the replication process. Mimics, it means that it is similar to the replication, but PCR is not replication. Because during PCR, only DNA fragment is amplified, not the whole genome. What does it mean? PCR is not replication. Please remember that. PCR only resembles replication, but it differs from it. First difference is that replication takes place only in living cell. It is also called in vivo and is a process in which DNA polymerase doubles the whole genome in cell just before cell division to make sure that both cells after division will have the same genetic information. Second difference between PCR and replication. In PCR, DNA is not replicated but amplified. It means that we are going to make a copies of a desired fragment of the genome. Summarizing, in PCR reaction, only one fragment of DNA is amplified in huge number of the copies. That's why we call this process amplification, not replication. It is conducted outside the cell. It is called in Latin name in vitro. So, as you now know, PCR is enzymatic amplification of DNA fragment in vitro with a length of several dozen to several thousand nucleotides, which lies between two regions of known sequence. In PCR reaction, there are carried out many cycles of synthesis of a DNA fragment using two primers which flank, it means surround, the specific segment of DNA. It allows to receive a large number of copies of specific DNA fragments. So we have many techniques based on PCR. For example, you see RT-PCR, RAPD, PCR-RFLP, AFLP, SSR, analysis of ITS regions, and many others. So what are the main components of the reaction? What we have to add to the tube, to the reaction tube, to make the reaction? The first is DNA. DNA must contain the sequence which we want to amplify. Second one, DNA polymerase, an enzyme that builds new DNA strands. And two PCR primers. Primers are short oligonucleotides between which the DNA sequence to be amplified is located. And nucleotides, the NTPs, they can be called as a building blocks. We can call it building blocks because from them DNA polymerase builds the new copies of DNA fragments. Now more details about the components of the reaction. DNA. DNA is also called template. Template contains the fragment of our interest to amplify. If this fragment is not present in the sample, reaction will be negative. And what about sensitivity of PCR tests? They are very high. In theory, only one DNA molecule containing sequences complementary to the primers should be detected and amplified during reaction. But unfortunately, sensitivity is lower, mainly due to the presence of DNA polymerase inhibitors in plant tissues, soil or water. That's why the purity and quality of DNA samples used in PCR must be very high. So you have to remember, dear students, that DNA has to be isolated very carefully with as low concentration of potential inhibitors as possible. Poor quality of DNA sample may result in complete lack of reaction results. Primers. Primers are single-stranded DNA fragments. They are also called oligonucleotides. They are short, normally typically from 15 to maximum 25 nucleotides. Primers show polymerase where to start amplification process. In PCR reaction, always two primers are used. So the primers, once again, please remember that the primer show the place on DNA strand from which enzyme polymerase has to start the process of building the new strand. Primers are individually designed for specific detection of desired DNA fragments. So you have to design your own primers. It means that primers are species-specific. For example, if you want to check the presence of two pathogens in one sample of DNA, we have to conduct two separate PCR reactions, each with different pair of primers designed for detection of pathogens which we look for. So primers flank DNA fragment to be amplified. Altogether, they define the final length of the double-stranded DNA fragment which will be amplified during PCR reaction. Primers bind specifically. This process is also called hybridization. So primers hybridize to both strands of DNA after the denaturation step. Each of them is bound to different strands of DNA. Primers have their own sequences which can be designed using special computer software, or you can find these sequences in scientific papers in which the authors publish them. So you don't have to know how to design primers with computer software. You have to just look for papers, scientific papers, in which the authors already designed the primers, and you can find the sequences of these primers in the paper, and you can copy these sequences, and you can order the primers specially designed for your experiment in your laboratory. The next component of the reaction, the mixture of four nucleotides, A, G, C, and T. Of course, these are elements from which polymerase will build the new strands of DNA. And Taq DNA polymerase. This is the enzyme which builds new complementary strand of DNA. Reaction starts from points where primers hybridized to DNA strands. Polymerase is thermostable and resistant to high temperatures. As you remember, this enzyme was isolated for the first time from thermophilic bacterium living in hot springs. The name is Thermus aquaticus, and therefore the abbreviation of this enzyme is called TAG, T-A-Q, T from Thermus, A-Q from aquaticus. So we call it this enzyme Taq DNA polymerase. Let's go to the details of PCR reaction. PCR is a cyclic reaction. The cycles are repeated from 30 to 40 times, and each cycle consists of 3 steps: denaturation, hybridization, and extension. The first step, denaturation. This is separation of double-stranded DNA, usually at 95 Celsius degrees. Under these conditions, single strands will be templates in the next step for the synthesis of complementary strands. Time needed for this process is about 1 minute. Second step, hybridization. It is also called annealing. Annealing of primers. Annealing means specific binding to DNA templates at lower temperature. Here I gave you the wide range of temperatures from 40 to even 65 Celsius degrees. Why the range is so wide? Because hybridization temperature depends mainly on the sequence of the primers. So the higher is the percent of G and C, the higher is hybridization temperature. Temperature also depends on length, on the size of the primer. The longer is the primer, the higher should be temperature, and the composition of the reaction buffer. This step takes from half a minute to 1 minute. And the last one, extension synthesis. DNA polymerase builds complementary DNA strands. Enzyme builds new DNA strands. The optimum temperature of this step depends on the origin of a thermostable DNA polymerase, and for Taq DNA polymerase the temperature is constant and it is always 72 Celsius degrees. And time you have to spend for this step is from half minute up to 2 minutes, and even more than 2 minutes. The longer is the product, more time is needed to build it by enzyme. So here you can see the graphic presentation of single cycle of PCR reaction. What we have here, please look at the slide. We have original double-stranded DNA fragment, so you have one strand and second strand, and the first step of single PCR reaction cycle is denaturation, 95 Celsius degrees, and we have separation of one strand from another strand. We have 2 single strands of DNA. Later, in the next step, which is called primer hybridization or primer annealing, we have lower temperature, and at this temperature, our primers, the primers are yellow here, and one primer, as you remember, we always have two primers in one reaction. The first primer will anneal to the one strand of DNA, which is produced after denaturation, and the second primer will anneal to the second strand of the DNA. And starting from the points in which the primers were annealed, in the third step, which is called elongation, enzyme polymerase starts to build new strands of DNA. So the blue one is the old, let's say old strand, and the new strand is marked as red. So this is new strand in this direction, and on the second strand, this is old strand, this is primer, and from this point going to the right, enzyme builds the new strand of DNA marked as red. So this is graphic presentation of single PCR cycle divided into three steps: denaturation, primer annealing, and elongation. After every cycle, enzyme doubles the number of desired specific DNA fragments. So after each finished cycle, you have 2 times more copies in this step than in previous one. So the number of DNA copies can be easily calculated using simple formula. 2 to the power of n. This is this formula, where n represents the number of cycles that are performed. So after 30 cycles, we should have 2 to the power of 30 copies of the fragment which we want to amplify in the reaction. So it would give us over 1 billion copies of this fragment. Now let me present the simple animation which will present you how PCR works. So it will show you how reaction goes, and I think it will help you to understand it better. So let's start the animation.
The polymerase chain reaction is a technique which allows one to amplify a specific region of the genome more than 1 billion-fold. A reaction is set up which contains a small amount of genomic DNA, 2 oligonucleotide primers which hybridize to either side of the region to be amplified, nucleoside triphosphates, the substrates for DNA synthesis, and the heat-resistant DNA polymerase, Taq polymerase. The reaction is placed in a PCR machine, which controls the temperature of the reaction. Amongst the many DNA sequences present in the tube, only the DNA located between the sequences that are complementary to the primers is amplified. This is referred to as the target DNA. The first step in the reaction involves denaturation of the genomic DNA by heating to 94 degrees centigrade.
Please look at the screen. Now this is the first step of the cycle. Denaturation, 94 Celsius degrees, and we have separation of the strands of DNA.
The reaction is then cooled.
Now we have the second step, primer annealing. This is the first primer and the second primer. Of course, each primer has its own sequence, and each primer will try to find the complementary fragment of DNA where the primer will anneal. And let's look what happens.
To 42 degrees centigrade.
The primers completely annealed, one to the one strand, second to the second strand, And this is the end of the primer annealing. And now we have the third step of the cycle, which is called elongation. And temperature will be 72 Celsius degrees,
allowing the primers to hybridize to the complementary sequences in the genomic DNA. The temperature is then increased to 72, the optimal temperature for Taq polymerase to synthesize DNA.
See, we have old strand of DNA, we have new strand. This is old, this is new strand. And our enzyme, Taq polymerase, built a new fragments of DNA. So we had one molecule, one copy of the DNA, and after this cycle we have two copies. And please observe what happens next.
And this completes the first cycle. Cycle 2 begins with the denaturation step, followed by the hybridization of the oligonucleotide primers, and then DNA synthesis by Taq polymerase. At the end of cycle 3, two of the products contain only the region between the oligonucleotide primers. At the end of cycle 4, 8 target molecules have been generated and 8 longer length molecules. The number of target sequences continues to grow exponentially until by cycle 30 over 1 billion target molecules and only 60 longer length molecules have been generated. The loading dye is then added to the reaction and the DNA fragments are separated by electrophoresis through an agarose gel. The amplified target DNA can then be eluted from the agarose gel for further analysis.
Okay, this is the end of the presentation. And finally, as you saw, we have to take out the tube from the machine after reaction, and we have to load the sample on the special agarose gel. And during the process, which is called electrophoresis, we have to separate the fragments of DNA which were amplified during reaction under electric field, and we can observe short flashing band which represents our product. Okay, let's continue. The reaction usually consists of 30 to 40 cycles. It takes, depending on the program and the type of the thermocycle, from several minutes to even more than 3 hours. PCR is performed in machines which are called thermocyclers. Thermocycler has to be programmed by setting up the number of cycles as well as duration and temperature of each individual reaction step. The reaction is carried out in a volume of 10 to maximum 50 microliters in special thin-walled plastic PCR tubes. Here you can see the examples of thermocycles. Here we have Biometra, MJ Research, and of course this is also Biometra but with 3 separate thermal blocks. But of course there are many other producers of these machines like Eppendorf, Biorad, and many others. Plastic tubes used for PCR have typical volume of 200 microliters, and you can use single tubes like this one on the photo. This is single small tube for PCR. To compare the size of this tube, you can see here bigger one. This is 1.5 milliliter. There is also possible to use groups of tubes for PCR reaction, and they are called strips. This is the strip. So they are connected together. This is 8 tube strip. 1, 2, 3, 4, 5, 6, 7, 8 tubes connected together, and also with special tube cap to close the tubes before the reaction. So you can use the tube individually, single tubes like this, or you can choose the strips. This is your choice. PCR reaction is conducted in very low volumes of reagents. They are measured in microliters, so you have to use special micropipettes to prepare such liquids. Of course, we can use traditional single-channel pipettes or multi-channel ones. So this photo represents single-channel pipettes, and this is multi-channel one, 8-channel. They can be mechanical or electronic, and pipettes are used with single-use disposable tips, which have wide range of volumes, from several microliter to 5 or even 10 milliliters. These are electronic pipettes, single channel and multi-channel. Here you see the use of multi-channel pipette for preparing strips of the tubes. These are the tubes as a strip. We have 8 tube strip here, 1, 2, 3, 4 strips. And the tips used for individual pipettes from very small volumes up to even 10 milliliters. So, electrophoresis. Samples after the reaction have to be checked by loading them on special gel. So results are observed under UV light, and in order to make DNA visible under this light, we have to use special staining dye. And this dye is added to the liquid gel, which is left to solidify. Staining dye intercalates into double-stranded structure of DNA, and dye DNA complex makes strong light emission under UV radiation. So you have to prepare special gel. You have to use agarose, you have to dissolve the agarose in a special buffer used for the electrophoresis. The most popular one is TBE buffer, and you have to dissolve this gel in hot buffer, and then you have to add special staining dye. You have to cool down the gel. You have to pour out the gel to the special tray. We have to use special comb, and this comb is used for making the wells in the gel, and in every well you will load separate sample after reaction. To make sure that all samples are on the bottom of gel in the TBA buffer, special loading buffer is added to each sample before loading. These are the samples mixed with special loading buffer. The loading buffer gives you the color, in this case it is the blue one, and makes the samples visible, and you are sure that you correctly loaded your sample into the gel. The loading buffer stains samples, gives them the color to make them visible in the gel. DNA is separated in the gel in TBA buffer under electric field emitted by power supply and goes from minus to plus. This is power supply, this is electrophoresis chamber, this is the gel, and you see the loaded samples, they are blue, and in this place, in this part of the chamber we have minus, and here we have plus. So electrophoresis goes into one direction, from minus to plus, so from here to here. So the effect of the electrophoresis is visible in the UV light emitted by the transilluminator. So this is transilluminator, the machine which emits UV light. You can see UV lamps under glass in this machine, and when you put your gel after electrophoresis on the surface of the transilluminator, you will see the flashing bands of DNA which under UV light emitted by the transilluminator. Of course, you can use special dark chamber. Inside this chamber you have the transilluminator. You have to open this door. You have to put the gel on the transilluminator and close the door, turn on UV light, and camera, which is connected with the computer, gives you the picture of the gel, and you see this picture on the screen of the computer. You can save this picture in electronic form on the disk of the computer. So techniques which are based on PCR are highly sensitive, and you can detect even trace amounts of DNA using PCR reaction. This is of great importance so we have now the examples of the use of PCR reaction. So let's discuss one by one. The first one, industrial quality research to identify harmful organisms in food and for detection of GMO in food. The second one, diagnosis of causal agents of plant diseases, bacteria, viruses, fungi in early stage of the infection when disease symptoms are not visible. So we can detect the pathogen even when we do not see any disease symptoms in the plant. Detection and identification of factors that cause human and animal diseases, for example in medicine and veterinary, and for example detection of viruses such as HIV in blood. The next one, confirming the species of pathogen in case when we are not completely sure if identification of this organism by conventional methods, for example microscopic, was correct. In this case, we have to isolate DNA from pure culture of microorganism and then conduct PCR with the specific pair of primers. Finally, we see the results on agarose gel. The next example, testing if some isolates have a particular genes, for example, coding the ability to produce mycotoxins. We can use PCR reaction to check the potential ability of some microscopic fungi to produce very toxic compounds which are called mycotoxins. The next one, determination of genetic similarities or differences between organisms of the same species or between different species. Forensic analysis, determination of the origin of traces of blood, hair, and others at crime scene. Analyzing DNA mutations associated with genetic diseases. Establishing paternity. Multiplication of DNA from Egyptian mummies and the remains of extinct organisms like dinosaurs, etc. Analysis of their origin, parentage, and so on, and many other areas. So, let's look at some examples of the results of PCR reaction. So the first one, I will show you some examples of PCR analysis and the results. Here you can see the results of PCR assay for determination of potential mycotoxigenic ability of fungi from genus Fusarium to produce mycotoxins from trichoticin group. This is detection of 3,5 gene which encodes special enzyme taking part in trichoticin's synthesis. This is the picture of agarose gel after electrophoresis, and there are visible clear bright bands representing fragments of DNA amplified during PCR reaction. So you see white bands, white flashing bands, and every band means positive result of the detection of this gene in the sample. So we have 16 samples, and every sample had the presence of this gene in the genome. What are the results? All tested DNA samples showed potential ability to produce mycotoxins. The presence of a band means that result was positive, and the lack of a band is negative result. See, the negative is here, 0, we have nothing. As you see, in the last well described as 0, we have no result. This is negative result where sterile water was added instead of DNA sample, so reaction is negative. In the first well here, you can see the ladder of a bands, and this ladder is called marker. Marker is used for determination of approximate size of PCR product. In this case, the size of the product is 545 base pair. So here we have band which represents 500 base pairs. This is 600 base pairs, and our product is between 5 and 600. And we have the product which size is about 545 base pair. On this slide, we have the example of detection of pathogenic fungus in wheat grain. Its name is Fusarium culmorum. So we had the samples of the wheat grain, and we have isolated DNA from these samples of the grain, and we have checked if we had the pathogen inside the grain of the wheat. The pathogen was detected in each sample, so we have once again clear visible bands, and you can see bands in the gel, and the product size is 550 base pairs. So it was the presence of the PCR results from our laboratory, but of course you can, if you want to detect some specific fungi in food, you can buy commercial sets of reagents for PCR reaction, and this is the example from the internet. And what we have here, as you see, there are kits for detection of, for example, Aspergillus niger, Botrytis cinerea, Cladosporium cladosporioides, Penicillium, Saccharomyces cerevisiae, plum pox virus, and Phomopsis viticola. So we have special kits of the reagents especially designed for specific detection of some fungus, viruses, or other microorganisms which can be detected in the food. And what food? Plant tissue and fruits. Tissue, plant tissue, fruits. Plant tissue and fruits. Wine and juice. Plant tissue, fruits and sap. And so you have special kits for the detection of the pathogens, and we have also the information where it can be detected: plant tissue, fruits, or maybe even wine or juice. And finally, I want to show you the example of the scientific paper in which we have the sequences of the primers which we can use for the detection of some specific fungi described by authors of the paper. So we have the example of the paper concerning PCR reaction for molecular detection of fungi producing aflatoxins. So the title is Detection of Aflatoxigenic Fungi in Selected Food Commodities by PCR. Authors published sequence of both primers. See here, we have the methodology of the experiment, and in this part, polymerase chain reaction, the authors published sequences of 2 primers used in this study. This is the first primer sequence, this is second primer sequence, and if you want to use them in your laboratory, you can order primers from specialized companies, so you have to copy the sequence of one starter, one primer, and sequence of the second primer, and you can order your primers from the special company, and what you have to do is to give them these sequences to synthesize the primers. And finally, when you buy the primers, of course, if you buy the reagents for the PCR reaction, you can repeat this reaction exactly the same as the authors did in the paper. In this case, primer sequences are here, and you can also see the picture of gel after electrophoresis. So see, after reaction, you should obtain one product which size is about 800 base pairs. So the authors designed one pair of primers which will generate the product, the fragment of DNA, which size is almost 800 base pair. So this is all for now. Thank you for listening to this presentation. The next one will concern real-time PCR reaction. Thank you for your attention. Bye.